Assalamu alaikum. I hope this video finds you in the best state of your health. This is an educational video based on amino acids, peptides and proteins. So let's start. Amino acids, peptides and proteins. These are my subtopics which I'm going to discuss with you individually. Now first of all, what we need to know is what amino acids are. In chemistry, we define them as organic molecules having two functional groups, the amino and carboxylic group attached to the same carbon, which we refer to as the alpha carbon. And in biochemistry, it can be defined as building blocks of proteins. Now the key elements of an amino acid are carbon and hydrogen because of their organic nature, oxygen because of the carboxyl group, and nitrogen because of the amino group. Now let's look at the general structure. This carbon here in the center is known as the alpha carbon, to which the carboxyl and the amino group are attached. The small r at the bottom is the side chain of an amino acid, which is different for each amino acid. It can be hydrogen, as in case of glycine, or methyl, as in case of alanine. So it is the reason for the diversity of amino acids. Now there are two types of amino acids, the standard or common amino acids, which are the building blocks of proteins, and the non-standard or non-protein amino acids, which are not part of protein, or after becoming the part of protein, they are chemically modified. Now let's look at the classification of standard amino acids. We classify the standard amino acids on three bases. The structural classification, as the name suggests, is on the basis of structure, the classification on basis of polarity, and the last one being nutritional classification. Now let's have a brief overview of the structural classification. It has further 6 classes. The amino acids with aliphatic side chains have carbon and hydrogen in their side chains. The hydroxyl group, this one is quite self-explanatory, having OH group in their side chain. The sulfur containing, these have sulfur in their side chain. The acidic amino acids are dicarboxylic monoamino acids. Dicarboxylic means they have two carboxylic groups. This also includes their amides. The basic amino acids are opposite of the acidic amino acids, being dibasic, meaning they have two amino groups and one carboxylic group. The aromatic amino acids have a benzene or non-benzene ring present in the side chain. The amino acids have amino group NH instead of amino group. Now let's look at their examples. Now for the sake of convenience, I have only included one example for each class. Now the first example here is glycine. Glycine is the simplest amino acid having sweet taste, and it has hydrogen in the side chain. The second class, hydroxyl group. The example is threonine with the hydroxyl group seen at the top. The sulfur containing group, the example is cysteine with sulfhydryl group present on the left side. The acidic amino acid, example is aspartic acid, and we can see the two carboxylic group present on the opposite ends and a single amino group present at the top. Moving on, we see the basic amino acid, example being lysine. The two amino groups are clear in the green color, and the carboxylic group is present on the right side. The aromatic amino acid, the benzene ring, is clear on the left side, and the example is of phenylalanine. Now the last here is amino acid. It includes only one member, the proline, and you can see here that instead of NH2 we have NH, the amino group present. Now classification on basis of polarity has further 4 classes: the non-polar amino acids, polar amino acids with no charge on R group, polar with positive R group, and polar with negative R group. Let's have a look at their examples. Now the non-polar ones are also called as hydrophobic. They have hydrocarbons in the side chain. I have included the example of valine, and you can see here that it has a hydrocarbon side chain. The second group is polar with no charge on R group. It has either OH or sulfhydryl group in their side chain. I have given the example of threonine. The hydroxyl group is present at the bottom. Now the polar with positive R group. This class includes the basic amino acids. You can see here the example of lysine, that the 2 amino groups have attained positive charge, NH3 positive. And the last class, the polar with negative R group. This includes dicarboxylic monoamino acid, which means that they will have 2 carboxylic groups. The example is of aspartic acid. Now we will have a look at the last classification, the nutritional classification. You are quite familiar with this one. The first class here is essential or indispensable amino acids, which means that these amino acids are very necessary in our diet as our body cannot synthesize them. The second class includes non-essential or dispensable amino acids. These amino acids we can do without in our diet as our body synthesizes it on its own. Now the last one here is new, the conditionally essential. These amino acids are simply non-essential amino acids which under certain conditions cannot be synthesized by our body and hence are needed to be supplemented in our diet. So now let's look at their examples. Now you can see quite clearly the essential amino acids having 10 members and the non-essential amino acids having 10 members. The yellow ones that I've highlighted are the conditionally essential amino acids. Now one thing to note here is that there is one conditionally essential amino acid present in the essential amino acid. As I've already stated that conditionally essential amino acids are required only under certain conditions. Now these conditions can be thermal injury or sepsis, and in case of arginine it is being an infant. Arginine can be synthesized in adults, but in an infant it is not synthesized, because of which it is required in the diet and thus is included in conditionally essential amino acids. Now the next topic here is the acid-base properties of amino acids and their titration curve. Now looking at the structure of amino acid, it is clear that it has both positive and negative charge. That is, it can accept and donate proton. So it means it has amphoteric nature. Depending upon the pH, it can behave as an acid as well as a base. When we have low pH, that is, we are adding acid to a solution containing amino acid, in that case the carboxylic group accepts the proton, because of which it will exist as a cation. When we have high pH, that is when we add a base to the solution, the carboxyl group loses the proton and thus appears as an anion. Now the term we need to know here is Zwitter or dipolar ion. It is a hybrid molecule containing both negative and positive groups. As we look at the structure of amino acid, it is clear that it is a Zwitter ion. The isoelectric pH is the pH at which a molecule exists as a dipolar ion and has no net charge, meaning it is electrically neutral. This I have already explained to you, that at low pH the amino acid will appear as a cation, and at high pH it will appear as an anion, but at the isoelectric pH it will appear as a Zwitter or dipolar ion having both negative and positive groups and is electrically neutral. Now for further understanding, we will look at the titration curve of an amino acid, being leucine here. Now as we look at the titration curve, as the process of titration starts, it is cationic at the beginning, but as the process proceeds, at the end at pH of about 9, we can see quite clearly that the amino acid loses its protein, loses its proton, and appears as an anion. So at low pH, leucine exists as a cation. At isoelectric pH, it exists as a Zwitter ion, and at high pH, it exists as an anion. Now let's move on. Now we have a very important topic: post-translational modifications of amino acids. We can define this post-translation modification as modifications that occur in the polypeptides during or after protein biosynthesis, that is translation. So it means that these are the modifications or changes occurring in a protein after it has been synthesized. We can also define it as proteome diversification occurring at protein level, that is after translation. Proteome simply refers to all the proteins synthesized by a genome. Now, post-translation modification can occur in two ways: by covalent addition, that is by addition of new groups, or cleavage of peptide bonds by protease. Protease is an enzyme which is responsible for cutting proteins. Now, what is the advantage of post-translation modifications? These modifications diversify the amino acids, and they can occur at any stage of protein life. Now let's look at examples of post-translational modifications. Now first example here is tyrosine with its derivatives. These derivatives, the T4 and T3, are thyroid hormones, and you can see here that both tyrosine and its derivatives are quite similar, only there is addition of the iodine group. So this is an example of post-translational modification by covalent addition. Now the second example here is by the cleavage of peptide bond. We are quite familiar with the protein insulin. It exists in inactive state, insulin prohormone having a single chain. After it has been cleaved by the protein cutting enzyme protease, it becomes active and now has two chains. Now the next topic, non-standard amino acids, is very closely related to the post-translational modification. Now we can define the non-standard amino acids as amino acids not incorporated by ribosomes. Now since we know that these amino acids are chemically modified after the process of translation, which means they are not synthesized by ribosomes. Now you can see here that a standard amino acid, after being modified, becomes a non-standard amino acid. Now, what is the importance of non-standard amino acids? They play very important role in protein structural regulation, and they are intermediates in metabolic pathways. Now, I've given here the example of urea cycle. The intermediates ornithine and citrulline are formed during urea cycle. They are non-standard amino acids. Now the next topic here is the peptide bond formation. The amino acids are linked together via peptide bond, represented as CONH linkage. Now the peptide bond is formed between carboxylic group of one amino acid and the amino group of the second amino acid. The reaction is referred to as the condensation reaction, meaning there is elimination of the water molecule. The resulting molecule depends on the number of amino acids present. If there are only 2 amino acids involved, then it will be a dipeptide, or if 3 amino acids are involved, then we will call it a tripeptide. Now let's look at the diagram. As we can see here that the first amino acid is eliminating its hydroxyl group from the carboxylic group, and the second amino acid is removing its hydrogen from the amino group, and a water molecule is eliminated, and as a result this linkage here CONH is formed, called the peptide bond, and the molecule resulting is called as dipeptide. Now what is the difference between a peptide and protein? Peptides are simply short chain of amino acids, whereas proteins are complex molecules made up of one or more long chain of amino acids. Peptides have limited number of amino acids, up to 50 molecules, whereas proteins have amino acids greater than 50. Peptides have less defined structure, that is, they cannot assume complex structures like primary structure, secondary structure, tertiary structure, and quaternary structure, whereas a protein can assume these complex structures. Now look at the diagram. The peptide simply consists of amino acids linked together by peptide bonds, whereas the protein is quite complicated, having more than 50 amino acids and having other interactions like disulfide interactions, ionic bonds to hold the molecule together. Now we will discuss the ionic behavior of peptides. As we already know that in order to form the peptide bond, the amino acids use their alpha carboxyl and amino group, but still they have a terminal carboxyl and a terminal amino group present, responsible for the ionic behavior of peptides. In some cases, the side chain R is also responsible for the ionic behavior. So peptides are referred to as the polyelectrolytes. What are polyelectrolytes? These are polymers whose repeating units bear electrolyte group. In the case of amino acid, the electrolyte group will be amino group and the carboxyl group. So the acid-base behavior of peptide can be predicted by the following: terminal carboxyl group, terminal amino group, number and nature of R group. As we can see here in this diagram, that in the center we have the CONH linkage, the peptide linkage, and on the right side we have the terminal carboxylic group, and on the left side we have the terminal amino group. Now, the determination of amino acid composition in protein. For this purpose, we will hydrolyze the protein. Since for the formation of protein, we utilize the process of condensation by eliminating a water molecule, so to liberate the proteins, we will introduce the water molecule, and thus we will be able to hydrolyze the protein. Hydrolysis can be carried out by acid treatment, alkali treatment, or enzyme hydrolysis. After the protein is hydrolyzed, we can use chromatographic techniques to determine the number of amino acids. Now we have structure of protein. As you are already familiar with this topic, I will just give you a brief overview. The primary structure is simply the linear sequence of amino acids consisting of peptide bonds between amino acids. The secondary structure is the twisting and turning of the polypeptide chain, and in order to hold the twisting, we have hydrogen bonds between the amino acids. The tertiary structure is the three-dimensional structure of protein having disulfide bridges, salt bridges, and hydrogen bonds. The quaternary structure has more than one polypeptide chains, and the interactions include hydrogen bonds, hydrophobic interactions, and ionic bonds. Now let's look at the diagram. As you can see here quite clearly that in the primary structure there is simply linear sequence of amino acids with peptide bonds. In the secondary structure, the amino acid chain twists and turns, and in order to hold this twisting and turning, we have hydrogen bonds. The tertiary structure is the three-dimensional structure of the protein, and the quaternary structure is the complex structure consisting of multiple polypeptide chain having many interactions in order to hold the molecule together. Now let's look at cytochrome C, which is a very important protein present inside our body. It is a hemiprotein, meaning it is iron containing. It is located in between the inner and outer compartment of mitochondria. Plays a very important role in ETC, electron transport chain, a pathway for ATP synthesis. It serves as an electron shuttle or transport carrier between protein complexes. It also has a very important role in apoptosis, which is programmed cell death. It is a water-soluble protein and peripheral membrane protein. As we already know, peripheral membrane proteins are temporarily binded to the protein either by attaching to integral membrane proteins or by penetrating the periphery of the membrane. As we can see here in this diagram of the electron transport chain, the cytochrome C penetrates the periphery of the membrane and behaves as electron shuttle or transport carrier between complex 3 and complex 4. Now let's look at the role of cytochrome C in apoptosis. Since cytochrome C is present inside the mitochondria, on receiving the apoptotic stimulus, it is released into the cytosol, where it causes the formation of apoptosome, which is a quaternary protein. This apoptosome formation then causes activation of caspases. Caspases are protease enzymes, as we have already discussed that proteases are protein cleaving enzymes. So this activation of caspases causes apoptosis. Now let's look at the diagram. As we can see here in the first one, that the cytochrome C is still present inside the mitochondria, but on receiving the stimulus, it moves outside the mitochondria and thus causes activation of caspases, leading to the apoptosis. I hope this video was informational for you. Thank you very much. Assalamu alaikum.